Hello everybody and welcome to the second to last lecture of the semester. So we're going to cover the immune system. Now, one of the things to remember here is that immunology is a huge field, right? And we could teach, actually you can get an entire PhD in immunology. So I'm going to give you a run through of some of the most important aspects of the human immune system in just 2 lectures, okay? There's so much information here. And if you ever, be an infectious disease nurse or work in the ICU, or nowadays, there's COVID floors in the hospitals and things like that. So this is going to be particularly relevant to that. Right? So there are two primary host defense systems that humans have, right? And they're very complicated, each one of them, and they work together. And sometimes not straightforward ways. There are 2 different systems, and there are 3 primary lines of defense. Okay? And this is a good overview that shows, some of the cellular defense mechanisms and kind of how they work in the body and what some of those members are. And we'll talk about some of these, but not all of these. Okay? So if we look at our, geez, I hate when it does that. I think every single lecture it does this to me. Yes, I do. I want to resume my slide. Okay. So we have our host defenses here. There are 2 primary branches of our immune system. We have our innate, non-specific, and then we have our acquired or specific, which is really our 3rd line of defense. This particular lecture, we're going to focus on the 1st line and the 2nd line defenses here, and then it breaks down further into here. And so this is a great kind of study chart. To give you a broad overview of each part of these lines of defense. So the innate and non-specific defense mechanisms is really we have a 1st line and a 2nd line defense, and so innate means something that is built in automatically. Okay, and so most of these defense mechanisms in the non-specific immune system are genetically hard-coded. And so they don't change over time, and they don't change with repeated exposures, right? So that's one of the key differences between our innate non-specific system and then something that we call the acquired and specific system, which is really our 3rd line and last line of defense. Now, your innate system is always on guard, no matter what. And like I said before, it does not improve with repeated exposure. And it also involves the inflammatory response and the phagocytic defense mechanisms. So, for example, that mucociliary ladder that we've talked about quite a bit is one of the key components of your innate non-specific defenses. Now, there are 2 major types of 1st line defenses. So we have physical barriers. And one of the biggest barriers, one of the biggest 1st line defense mechanisms is this physical barrier, which is our skin. And we'll talk about the skin here in a moment. But there are also chemical barriers like tears and saliva and sweat. There are some genetic barriers, such as maybe not having the receptor for attachment or something like that, or a mutated receptor. There are also some physical barriers, such as your thick skin, right? Cilia, which are part of that mucociliary ladder, the mucus secretions, which is kind of like a fly trap, for example, or some other types of secretions like saliva and tears, right? And so here's just an example of some of the chemical barriers that you may see that are part of the non-specific defense in different parts of the body. Now, the one thing that is important here to note is that in this non-specific system, it operates regardless of invader. So think about a castle, right? And a castle has lots of defense mechanisms, right? And like an old medieval castle, right? What are some of the physical barriers that a medieval castle may have that prevents people from attacking it and taking it over, right? So, okay, a moat, right? We surround the castle with this wide thing of water so people can't get in, right? It doesn't matter what kind of person it is. It doesn't matter what it is, whether it's a cart or a horse or a person. They can't cross that moat, right? Or what about the walls, castle walls? It doesn't matter who's trying or what's trying to get into the castle. It's a general defense, right? Physical barriers. And then there are chemical barriers, right? So, for example, back in the medieval times, people tried to get in through the main castle gates, and people above the gates would pour like hot tar and use fire and things like that. Like, those are chemical barriers. It doesn't matter what gets caught on fire. That thing that gets caught on fire, regardless of what it is not going to make it through the castle door, right? So these are non-specific defense mechanisms. There are also some cellular defense mechanisms. And we'll talk about some of the granulocytes and agranulocytes and the things that they have. And then there are also molecular defenses, right? And these are specific proteins which are produced by very specific genes that encompass some antibodies and other antimicrobial compounds. This is part of the genetic component of innate immunity. Now, we mentioned the skin already, the epidermis, which is the outer portion, and the dermis, which is the under portion. Now, the epidermis is completely renewed, every 20 days or so, 28 days. And the top layer is dead and contains a protective protein called collagen. It also contains keratin, right? So the outer portion of your skin is also made up of keratin, which is a waterproof and protective protein as well. Now, many of the external portions of your body are what we call mucous membranes, right? And they are sticky. And in that mucus, there are various defensins, things that are secreted in there, such as antibodies like IgA and IgD, and things like lysozyme, right, which break down peptidoglycan in bacterial cell walls. Remember? Right? There are some acids and salts that your body produces: saliva, perspiration, stomach acid, right, bile in your tears. And then there's that lysozyme protein that we talked about that we stole from viruses. And so this is just an example of where we might find that. Now, urinary tract infections are much more common in individuals that are unable to empty their bladder, right? Or that don't pee enough, right? So if you hold your pee for long periods of time, chronically, for a long period of time, that can lead to an increased susceptibility of infection, right? So typically your bladder is sterile, right? It's considered to be inside of your body, but there is your ureter and your urethra, right? So your urethra is a portal from the outside environment to the inside, but usually it's kept very tightly closed. And if bacteria are able to get up and through, which they can, right? If you don't pee enough, they can make it all the way up into the bladder, which they can cause an infection. So urination helps remove bacteria which have made it up any portion of the urethra. Right? Now, typically, females are more susceptible to bladder infections than men, right? Because they have a shorter urethra, usually, than most men do. Now, when we talk about the physical barriers, right, the normal flora of the skin inhibits a significant amount of potential pathogen microbial growth. Okay? Now, we also see that in areas where we have a hot, humid climate, like the tropics and things like that, we see an increased rate of skin infections. And again, that has to do with the fact that there's more moisture than in the air, which allows more organisms to grow. And it also allows the organisms that are present on your skin, as part of your normal flora, to grow more than what they should. So we've got the epidermis here, and then we've got the dermis below it. And there are sebaceous oil glands and eccrine sweat glands and all kinds of things that will help keep the surface of our skin relatively clear of microbial invaders. Now, the skin itself is your largest organ, right? And we pretty much talked about that. And the deeper layers of your epidermis contain some of these stem cells, as well as some phagocytic cells, which we call dendritic cells, and we'll talk more about them as we turn. And then we also have collagen in our dermis, which creates kind of this really tough, rigid structure. I just realized that strength is misspelled here. And some of the pliability that's concerned with our skin. Now, one of the primary dendritic cell types that we see in our skin are the Langerhans cells, right? And this is the second line defense mechanism of our skin. These Langerhans cells have really long kind of like appendages, so that this larger brown portion, which is specifically stained in this skin slide, is the central portion of the cell. And then it extends it's almost like an amoeba. It extends its arms around the cells of the lower portion of the epidermis. And it moves, right? It can squeeze in between these cells and kind of move around like an amoeba would. And what these cells are doing is they are looking for the presence of anything that they determine to be foreign, right? So things that aren't you, right? And that's one of the big things about the immune system is it always has to be able to recognize you from everything else, which is actually a very difficult thing. And we'll talk more about that in the next lecture. So these Langerhans cells are kind of roving your skin, and they are looking for bacteria that have made it through, viruses, cancer cells, all kinds of things, so that they can then run and tell your other third line immune defense system, your specific immunity, hey, we've got invaders here, let's mount an attack, right? So this is a primary interactor in the skin between the skin's first line and second line defense mechanisms. We talked a lot about the mucous membrane and the cilia already. We've also talked about how they line the respiratory, genitourinary, and gastrointestinal tracts, right? And they really make that mucociliary ladder. As far as chemical barriers are concerned, sebum, which is oil that's secreted from the sebaceous glands of the skin, contains lots of antimicrobial peptides, and there's a slightly lower pH there, which helps inhibit the growth of many microorganisms on the skin. There's also lysozyme, which is present in tears, your watery sweat, as well as saliva, and that helps break down a lot of microbes there. Your gastric acid, Ryan, is highly acidic. And then these antimicrobial peptides, which I mentioned, which are in sebum, are also found in other secretions like cerumen and a little bit in sweat as well. Now, one of the most important yet growth-limiting micronutrients that bacteria have to make sure they have a source of is iron, right? Iron is found in the human body in relatively high concentrations as far as a bacterium is concerned because we use iron in our blood, right? It's our primary oxygen binding component of hemoglobin, right? And so if our body realizes that there is a microbial attack, right, it can increase the secretion of a special protein called a siderophore, right? And siderophores are enzymes, or not really enzymes, but they are proteins which their sole job is to bind up and lock away any iron which is floating around. Okay? And so if we secrete lots of siderophores, it will kind of remove these iron molecules from being able to be reached by bacteria, which will severely cripple the growth of most bacteria. We talked a little bit about sebum and the fact that it has a lower pH. It contains a lot of different types of organic acids, and most sebum will inhibit the growth of certain pathogenic fungi. And then staph epidermidis and Propionibacter acnes, right? This is also part of your normal flora, but in certain cases it can cause acne, right? Excrete certain fatty acids which are toxic to many different types of gram-negative organisms. Right? Lysozyme, right, breaks down bacterial cell walls, specifically peptidoglycan. It's the most effective against gram-positive organisms. And in humans, tears have the highest concentration, right? So lysozyme is very important in preventing ocular infections, right? And to a lesser extent, it's also present in blood, sweat, urine, and in actual body tissues themselves. Right? So we kind of talked here about sebum. And then in lysozyme here, we've got our lacrimal glands, which are on the lateral portion of your eye, right? And they secrete tears, and the tears wash over into your eye, and then the tears are collected in these lacrimal canals, and then it's all collected into the nasolacrimal duct, which then goes into your nasal cavity where you swallow. Now, our second line of defense is our cellular defense, right? And the cells that we're going to talk about in second line defense mechanisms here can also play a role in the specific immunity in our third line of defenses as well. So the second line, this is where the non-specific and the specific immune system starts to overlap a little bit. Right? Now, in your blood, you've got two main parts, right? You have a liquid part of your blood, which is called the plasma, and you have the solid portion of your blood, which is cells and cell fragments, which are mainly platelets. Right? Now, the liquid part of your blood is called plasma unless it's allowed to coagulate. Right? If the blood is allowed to coagulate and then you spin it down, then the liquid portion, which now has no clotting factors, is called serum. Right? So the different colored tubes that you may see or collect when you're drawing blood or having blood drawn have different chemicals in it which either allow clotting to occur, like a red top tube, or that prevent clotting from occurring, like a purple top or green top tube, right? And different blood tests require different types of blood preparation, so either serum or plasma. Now, within the solid portion of blood, you've got different populations of different cell types, right? So you've got your erythrocytes, which are your red blood cells, your RBCs, that make up the majority of the cellular components of your blood. And they're the ones that move all the oxygen around, right? And contain all the iron and hemoglobin. And then you've got the leukocytes, right? Cyte means cell, leuko means white, so these are the white blood cells, right? Now, these white blood cells are the ones that are the most important, and they participate in the non-specific and the specific defense mechanisms and systems, and that's kind of where we'll focus. And then the other portion are platelets, and platelets themselves are actually not cells, they are fragments of cells. There are some specialized cells in your bone marrow which are called megakaryocytes, and these break off pieces of themselves, and that's what platelets are. Now, in the leukocyte world, there are two major divisions. There are the granulocytes, also called polymorphonuclear leukocytes. Right? So let's break down that word. What does poly mean? Well, poly means many. What does morpho mean? Means shape. And what does nuclear mean? Means nucleus. Right? So these are white blood cells, leukocytes, which have a many-shaped nucleus. Right? Okay. So granulocytes typically have a granular cytoplasm. There are tiny little vesicles inside of the cytoplasm which contains chemicals. And these chemicals can be released to the environment when the cell is stimulated by some kind of external signal. And they stain differently depending on what chemicals are contained in these vesicles. Right? And we'll see that in a minute. Right? And the counts of these granulocytes can be diagnostic. Certain conditions you have one type elevated, or other conditions you have another type, and so on and so forth. The other type of leukocyte which will be important are the agranulocytes, and they lack these granules, and they typically have a relatively smooth and round nucleus. Right? And we'll talk about those later. Those are the monocytes usually, and the lymphocytes. Now, in the granulocytes, basophils, right? Typically we see some of these in the bloodstream, but usually very low concentration. Many of these are found in the tissues. And when white blood cells squeeze out of your blood vessels into surrounding tissue, that's called diapedesis, right? Diapedesis. Or sometimes you'll see it called extravasation. They are leaving the vessel system, right? And most basophils we find in the dermal and the epidermal regions of the skin, and sometimes in organs as well. Now, these basophils are super important in inflammation, and they are responsible for initiating certain inflammatory and allergic reactions. And the purple granules that you see that are stained here, and basophils typically look purple and granular, right? This is actually an agranulocyte here, right? And so they release histamine, right? And so if you have a bug bite, and that bug bite becomes itchy, right? You had some allergic response. That means the basophils in the area around that bug bite released a crap ton of histamine, which is causing your itchiness and redness and swelling that are associated with an allergic response. Right? When you take Benadryl, for example, an antihistamine, right? It blocks the release of histamine from these basophils, which prevents then the allergic reaction or the localized inflammatory response to happen. Now, eosinophils, they have red staining granules, right? You can see the multi-lobate nucleus here. Eosinophils are usually associated with an increase in number during parasitic infections, and so they'll usually look for eosinophilia in a white blood cell count or a diff count when they're looking for a parasitic infection of some sort. Eosinophils are also important in the inflammatory response, and there are many autoimmune disorders which are related to eosinophilic dysregulation. Right? And a lot of times these eosinophils will detoxify foreign substances, and they can release lots of different degradative compounds, so compounds that break things down, such as the peroxide anion and the superoxide radical, right? Remember, these are dangerous forms of oxygen that most of the time our body has to detoxify. But some white blood cells and other cells in your body can actually use these dangerous forms of oxygen to break chemicals apart or even kill microbes. Now, neutrophils are the most common granulocyte that you'll see in circulation. They are often phagocytic, so they're the ones that eat foreign things. And they're usually the first cells at the scene of the crime, right? These are your typical traffic cops, right? They are the first ones to respond to a scene. They assess the situation, and they relay information to those that are higher in their chain of command, right? They also will mount something called complement. This is a part of the molecular second line of defense in the non-specific system, which we'll talk about in a little bit. Right? They represent 60 to 7% of all circulating white blood cells, and they are released from the red bone marrow in the bloodstream, or into the bloodstream. And they can also enter tissues via diapedesis or extravasation, right? So most white blood cells have the ability to leave your blood vessels when they sense an issue in surrounding tissue. They secrete a lot of things. They can secrete lactoferrin, which is a siderophore. They can also create or secrete proteases, which break down tissue and bacterial proteins, and they can also increase permeability increasing protein or secrete BPI, right? And this protein, when secreted, will increase the permeability of blood vessels, right? In other words, it will make them a little leaky, and so bacterial cells will have the ability to leave the blood vessels easily or more easily to get into the tissues. Agranulocytes, such as monocytes, they have one typically horseshoe-shaped nucleus, and they're usually very big. And they're agranulocytes, so they have a clear blue cytoplasm. They migrate into tissues, and sometimes they just wander. Right? Sometimes they can be fixed. In other words, the same cell cannot be removed or go away from a specific tissue type. Now, once they establish themselves in a particular tissue area, oftentimes they will differentiate into a macrophage. Right? So monocytes have the ability, when activated, to turn into macrophages. And a macrophage is a professional phagocytic cell. Their entire job is to find a foreign invader and kill it by eating it. Right? And these macrophages will then take the bits and pieces of whatever it is they ate, and we'll use that to activate the specific immune system. We'll talk more about this in the next lecture. Lymphocytes are the next kind of largest number of cells that you'll see in circulating red blood cells. And the lymphocytes are subdivided into two major categories, which then each have their own subdivisions. The T cells and the B cells. Right? And the T cells are important for kind of cell-mediated immunity, and the B cells are important for humoral immunity, which is antibody production. Right? They circulate in the blood, and they are the primary cells that we're going to discuss in the next lecture that are the main players in adaptive or specific immunity. So this is a good kind of overview chart that shows all these different blood cells. Right? Now, we haven't really talked much about hematopoietic cancers in this class. We did talk a little bit about cancer, but different types of cancers result from mutations that occur in different subsets of blood stem cells. Right? So, like AML, right, acute myeloid leukemia, or CML, chronic myeloid leukemia, occur when mutations happen in maybe even just one of these myeloid stem cells, right? And that then causes overproduction of all of the granulocytes generally, and occasionally the monocyte, but mostly neutrophils, basophils, and eosinophils. Whereas lymphoblastic leukemias, both acute and chronic, so ALL or CLL, arise from mutations that typically occur in a lymphoid stem cell, right? And that will result in dysregulation in B, T, and NK cells. Right? So there are blood vessels in the body, right? And these are the primary ways in which your red blood cells migrate throughout your body. But there are also lymphatic vessels. And the lymphatic system is one of the primary systems that white blood cells use to traverse the body. And lymphatic ducts directly connect the arterial and the venous side, as well as to all the tissue as well. And the lymphatic system has very leaky walls, and so liquid can very easily diffuse in and out of the lymphatic system, the same with cells. So cells can easily move in and out of the lymphatic vessels and then directly into the bloodstream. So this lymph system is really a highway that's specific for the immune system. Now, phagocytic cells, which are represented primarily by macrophages and neutrophils in the human body, they are the primary scavengers, right? They are looking for anything that is foreign, anything that can be identified as not you. And this is at the molecular level. So your immune system can recognize your cells from any other cells in the world, right? Based on the fact that you have a very unique genome, right? And your genome means that you have a very unique proteome. And so your proteins are different at the molecular level than another human being's proteins. And again, that's the basis for host graft rejection, right, in transplants. These phagocytic cells are found in blood, in tissues, in your liver, right? And generally the process of phagocytosis is the process of ingesting and digesting foreign particles. And this could be dead cells, cellular debris from them being killed by something else, or from a virus destroying them, and also by directly ingesting invading microbes. And you can see this is not a human cell, but it's a different cell, and it is ingesting yeast. So these are yeasts, and it's phagocytizing the yeast, binds to the yeast, and brings the yeast inside of it, and then it will digest those yeast cells. Now, the general way that phagocytosis occurs is chemotaxis. First, right? So we talked about chemotaxis and we talked about bacteria. What is chemotaxis? Well, it's the movement of a cell up the concentration gradient of some chemical, right? So how do white blood cells know that you have an infection in your toe, for example? Well, the infection starts in your toe, and as the local cells that are in your toe, the local members of the non-specific immune system union, right, in your toe, they recognize that there's some invaders, and they start secreting chemicals into your bloodstream that originate in your toe, right? So these chemicals will diffuse away from your toe, right? And some other cells in your body, phagocytic cells, for example, will recognize that there's a phone call being made from your toe. And they will follow the concentration gradient of the chemicals being released by the cells of your toe. And as they get closer to your toe, what's going to happen to the concentration of those signals? They increase. So they are using chemotaxis very similarly to the way a bacterium would to find the exact location of where these phone calls are being made from. Right? And once they get there, they will find the bad guys again through chemotaxis, and they will stick to them, right? Adhere to them. Then they will bring them inside and ingest them. Then they will kill them, and then they will eliminate the particles that have been broken up by ingestion and killing of these things, right? And this is really important for the specific immune system, which we'll talk about here in a moment. All right, so now let's talk about the acute inflammatory response, because really one of the major underlying processes that is so important to initiating an immune response is an inflammatory response. So let's talk about kind of the evolution of this response and some of the things that it does and what's actually happening in the body when it happens. Okay? All right, so let's say you get a puncture injury, right? Splinter, right? Splinter from the outside environment. Splinter brings in bacteria from the outside environment, right? And what else happens here to cells of your skin when you puncture or slice it? Yeah, you're cutting them open, right? Now, one of the coolest things about your immune system is that it can recognize certain molecules very specifically. And a single molecule can act like a phone call. And that molecule can cause other cells in your body to release more molecules, which can call and make more phone calls, and so on and so forth. And so there are certain chemicals that are only found inside of a cell, right? And so as long as your cells remain intact, the phone call never gets made. But if for some reason the cell gets destroyed and breaks open or lyses, those molecules are then released and the phone calls can be made, right? We call these damage-associated molecular particles, right? Or DAMPs, D-A-M-P. Also, there are specific molecular patterns that are associated with this, and sometimes it's damage-associated molecular patterns. But these DAMPs are then recognized by local cells, and they can make more phone calls, okay? So you have damage to cellular tissue, right? You have the release of DAMPs, D-A-M-Ps, and you have the invasion of bacteria and viruses and whatever else happened to have been stuck on that splinter, right? So now you've got these bacteria which are also leaking chemicals, right? They are secreting things and leaking things. And so cells are also going to recognize those signals. So now we have some local cells, and they are secreting these chemicals. And these chemicals are then going to be recognized by white blood cells, which are circulating in the immune system, right? They are going to adhere to the blood vessels that are immediately adjacent to where the highest concentration of these chemicals are. Right? These leukocytes, these white blood cells, are going to squeeze between the cells of the vessel, right? And it's called diapedesis. Right? They are going to be attracted by all of these molecular phone calls, so to speak, right? And they are now going to begin the phagocytic pathway. They are going to begin eating bacteria that are in the local area. They're going to begin eating some of the cell debris in the area to help try and clear the wound. They're going to attack the splinter if it's kept in there, right? They're going to attack everything, right? And so that's the basis of chemotaxis here. And that's how chemotaxis begins the phagocytic response and the inflammatory response. So we've got a tissue injury, we've got the introduction of pathogens here, we have these chemical signals, and these chemical signals are going to cause these white blood cells to attach to the vessel. That's going to cause these white blood cells to secrete chemicals which are going to cause the vessels to dilate, right? If the vessels dilate, then the blood flow in that region happens to occur quicker, faster, you get more blood, which more blood means more white blood cells, right? They're also going to increase vascular permeability, which allows these white blood cells to squeeze out a little bit easier, right? Then once they're out of the tissue, they are going to start eating everything up, and they're going to initiate an inflammatory response, an immune response, and a wound closure response or a healing response. So we just talked about all this stuff, right? Chemokines, this is a general category of signal molecules that are secreted by all different kinds of cells in the body. And the primary group of chemokines that many of the white blood cells secrete, they're called interleukins, right? Leukins means white, right? So these are molecules or signals or phone calls, if you will, that occur between different white blood cell types. And different interleukins are different messages, right? And so these chemokines that are produced, right, they are released by these phagocytes, and they attract more phagocytes. So these phagocytes are making phone calls to other white blood cells, right, by secreting these interleukins or chemokines. So once these phagocytes arrive to the scene of the crime, they're going to attach, right, to whatever it is they're trying to eat. And if it's a microbe, then to the microbe. Now, some bacteria, if you remember, have virulence factors, like capsules. And that makes attachment extremely difficult. We've talked about Streptococcus pneumoniae, right? Remember the Griffiths experiment. And then Haemophila influenzae type B has a very thick capsule which actually inhibits phagocytosis. Right? Now, if the organism is ingested, right, has been taken up, then the cell membrane that surrounds the microbe, it forms what we call a phagosome, right, or some kind of a food vacuole. And then that encapsulated bacteria sometimes can interfere with the formation of a phagosome, right? Such as pneumococci, which we talked about. Right? Now, digestion occurs when the phagosome, which is now inside of the microbe, fuses, or inside of the white blood cell, fuses with the lysosome. Remember, the lysosome is that garbage disposal organelle that eukaryotes have, and inside of the lysosome there are acids and other proteolytic enzymes that degrade things. And so when the phagosome fuses with the lysosome, then that means everything that's inside of the phagosome will get destroyed, right? Digested. Most bacteria are killed within 30 minutes of phagosome lysosomal fusion. Okay? There are lots of, basically the phagocyte can use some of the breakdown products, right, into small molecules, and the phagocyte can then use that for energy, like sugars and things like that, right? And there are disinfectants, so to speak, that your body can make, and we kind of talked about the use of toxic oxygen, such as hydrogen peroxide, and even hypochlorite, bleach. Your body can make the anion, which helps kill microbes. This is a good graphical overview of what's going on here. We have chemotaxis to the area where the microbes are causing an invasion. We have adherence to the microbes. We have ingestion by the phagocyte of the microbes. Here's our phagosome. And then we have fusion. We have phagosomal lysosomal fusion, right, into something called the phagolysosome, where we have digestion. And when the bacteria is digested, some of these pieces can be removed here and used for other things, but then a lot of these pieces are eliminated by exocytosis. Right? I mean, it's the same as when you eat a candy bar or a Whopper Junior, right? Anything like that. Now, there's a lot of dying microbes constantly. Your immune system is under constant attack. And why are you not sick all the time then? So your immune system has evolved to be extremely efficient. And it has to be because of the constant attack, right? You are constantly under bombardment and your immune system never sleeps. It's constantly working even while you're sleeping. And that is one of the reasons why you never get sick. But if something happens to your immune system and you become immunocompromised in any way, that can severely cripple your ability to prevent infection, then you get sick all the time. Right? If you are one of those people, which unfortunately a small percentage of the population are, that is always sick by something, you must have some kind of a hole in one aspect of your immune system. Right? Something that a very particular type of microorganism can use to get through your first and second and even third line defense mechanisms. Now, microbes themselves have also evolved a way to defend themselves against your immune system, right? There's this molecular arms race that's happening constantly between you or your immune system and microbes themselves, right? So the microbes are always developing new weapons to use against our defenses. Right? So many microbes can resist or can resist digestion by encapsulation, right? And so in macrophages, for example, the capsule allows the microbes to actually live within the macrophage, and these capsules can prevent lysosomal fusion. Right? For example, Yersinia pestis, which is that gram-negative rod that causes the bubonic plague, has the ability to resist macrophage destruction. Right? You can also prevent the fusion completely by secreting things. And so Mycobacterium leprae actually can secrete different things inside of the phagosome, which prevent the lysosome from fusing with the phagosomal vesicle. And many microbes can secrete exotoxins, such as the streptolysins we've talked about, and another class of these chemicals, these toxins, these exotoxins, called leukocidins. And leukocidins are an interesting class of proteins. They are secreted by lots of different microbes. And these exotoxins specifically target white blood cells. They have evolved to destroy only those cells that are seeking to destroy the bacterial cells themselves. Now, we talked a little bit about chemotaxis, right? And the acute inflammatory response is typically your body's way of dealing with tissue damage, right, or a localized infection. Most of the time, an inflammatory response does more good than harm, right? Chronic inflammation is never a good thing, and we'll talk a little bit about that in a few minutes. But a typical inflammatory reaction is characterized by 4 cardinal signs: calor, which is called pyrexia, which is an increase in localized temperature, right? Rubor, which is called erythema, which is a localized redness, right? Tumor, which simply means swelling or edema, and then dolor, which is typically some type of analgia, which is pain, right? And each one of these things is caused by a different underlying physiological process. Right? Now, erythema, which is the redness in your skin, is typically caused by vasodilation, right? So we saw that chemotaxis where these neutrophils or macrophages or whatever happens to be there first are secreting this vascular permeability increasing protein, which dilates the blood vessels that are local, and that increases the blood flow to the area, which allows more fluid to seep out. So we're getting red blood cells that occasionally leak into the surrounding tissue, and that's causing the redness we see. Okay. Now, red blood cells leak into this area. Oh, I just said that. And then there is cytokine release from these white blood cells and the damaged tissue, which is going to be kind of like a positive feedback loop here, which is going to further increase chemotaxis and vasodilation, which increases localized redness. The swelling is predominantly caused by that vasodilation, right? And the cytokine phone calls that are being made from the area where the tissue damage or the infection is present is going to be increasing that capillary permeability, which is going to then have a positive effect or positive feedback effect on the vasodilation, which is really causing a lot of this edema and redness. Right? Now, if it's an active bacterial infection and your immune system mounts a good enough attack, there will be an accumulation of dead or dying members of the war, right? And so there'll be dying white blood cells, dying bacteria, and these will build up. And if they build up, oftentimes pus will accumulate. And that's really what pus is. It's the accumulation of white blood cells, both dead and some alive, as well as any damaged tissue debris and bacteria which may be in the area. So again, all of these things are increasing capillary permeability. So the injured tissue releases the damps, right, as well as cytokines, and these are going to call mast cells and basophils to the area, as well as macrophages to the area. And then the macrophages are going to secrete another chemokine called tumor necrosis factor alpha, or TNF alpha, which is also going to increase capillary permeability. Histamine is going to be secreted by mast cells and basophils as they react to the injured tissue, which is also going to increase capillary permeability. So you've got these three major things which are all working together, and they are also positively influencing each other's signals to ensure that enough cells get to the area of damage. Now the pain is important, right? Pain is alerting you to injury. If there are some genetic conditions which cause the particular individual to not be able to feel the sensation of pain, and it's very dangerous for these individuals because pain alerts you that there is damage currently being done or damage has already been done to a portion of your body, right? And these injured tissues typically release some type of a cytokine, and these cytokines can stimulate pain receptors. Prostaglandins are the big ones. These are cellular regulators which are connected to pain receptors, and these prostaglandins will also help mediate an inflammatory response. Right? These prostaglandins will contract smooth muscle. Now, why is that important, right? Well, you cut off a finger, right? Well, why would you want to contract all the smooth muscle in the area of the tissue damage, right? Well, smooth muscle is your arterial cell walls, and if you contract those, you can slow down bleeding, right? So prostaglandins will help contract smooth muscle in the area, and the prostaglandins themselves will increase the effects of some other pain-inducing compound called bradykinins. So if you have bradykinins being secreted as well as prostaglandins, you'll have an amplification of the pain in the region. Now you've also got swelling of tissues, right? And so you have free nerve endings that are oftentimes found in areas of damage, and the swelling of the tissue puts pressure on the nerves, which then increases the feeling of pain. Right? Now, there are lots of ways to mitigate the pain, right? So that's we can take NSAIDs, right? Non-steroidal anti-inflammatory drugs like aspirin, for example. And aspirin works directly by suppressing the release of prostaglandins. So if we suppress the release of prostaglandins, we will not intensify the effects of bradykinin, we will lower the pain, right? Now, if you are actively bleeding, should you take aspirin? No, right? Aspirin will prevent clotting, and it will prevent the contraction of smooth muscle, which slows the bleed, right? So knowing a little bit about the mechanism of the different drugs that you're giving your patients or that are prescribed to your patient will help you understand whether or not a patient should get the medication in certain situations, right? Now, there's also Tylenol. Tylenol is not an NSAID, it's its own kind of thing, it's acetaminophen. Now, it directly inhibits the inflammation pathway, right? Now, aspirin is an or what we call an analgesic, right? It suppresses pain. But aspirin does not do much for inflammation. Okay? Acetaminophen will inhibit an inflammatory response, but does less against pain unless the pain is directly due to inflammation, right? So understanding the effects of these different drugs can help you figure out when you should take one, right? If you cut yourself, right, taking acetaminophen will not really help your pain, but aspirin may. But taking aspirin, if you're actively bleeding, right, will cause you to bleed more. Right? If you sprain your ankle, for example, most of the pain that's associated with the sprain is due to the inflammation that is caused by the damage to the tissue that you did when you sprained. So taking acetaminophen would benefit you, one, by reducing the inflammation, which then reduces the pain. But taking aspirin would do not much, right, in that situation. So kind of understanding the mechanistics of these different pain relievers can help you determine when you should take one. Right? So this is how all of these things kind of work together. So when we have an acute inflammation response, right, the localized increase in temperature in the area is caused by vasodilation, which means more blood's going to the area, which means more of your 37-degree body fluids are getting there, which increases the localized temperature. The vascular increase in vascular permeability is allowing more red blood cells to leak out, which is increasing the redness, as well as more liquids to leak out of your vascular system, which is causing the localized swelling. Right? The damage to the tissue itself increases the release of prostaglandins and bradykinins, which directly stimulate the nerve endings there, as well as the increase in edema of the area also puts direct pressure on free nerve ending pain receptors, which are going to increase the localized pain in the area. So all of these symptoms of an acute inflammatory response are all intertwined, and they all kind of start with the increase in vascular permeability and the vasodilation, right? brought about by the secretion of these different chemokines. Now, a fever itself is a systematic increase in body temperature, and this happens at the central nervous system level. So typically the hypothalamus has to reset your metabolic rate, and a substance which elicits a fever is called a pyrogen. Right? There are exogenous pyrogens, so these are things that are secreted from pathogens, which can alert your body that there is an infection. There is also endogenous, right? And these are released by certain white blood cells, and these white blood cells are telling your body, hey, I found a pathogen, let's increase our temperature a little bit so that we can maybe slow down the pathogen's growth, right? And that's an endogenous pyrogen. Right? Now, interleukin-1 is probably the most potent endogenous pyrogen, and that really is what causes the hypothalamus to release prostaglandins, and will increase the temperature, and will also constrict blood vessels. Right? Now, there are lots of benefits to a fever. You increase the localized temperature, you increase the movement of enzymes, which increases biochemical rates, which increases tissue repair rate. It can denature the invader's proteins and slow down their growth. It can increase some of the important immune-based chemical reaction rates in your body. And it can also increase the activity of some interferons, which are antivirals. Right? Now, leukocyte endogenous mediator, LEM, is something else that is secreted by certain white blood cells, and it will help increase the fever, and it will actually also increase lactoferrin and transferrin secretion, which lower levels of iron available in the plasma. So we talked about all of that kind of thing. Right? Now, aspirin and acetaminophen are both antipyretics. So if we lower the localized inflammation, right, or even total body inflammation, which is acetaminophen, we'll lower the amount of prostaglandins and other chemokines such as interleukin-1, which are secreted, which will help lower the temperature. Aspirin is a prostaglandin inhibitor, so if we prevent prostaglandins from being secreted, then that will lower the temperature. So they're both antipyretics, but they do it in slightly different ways. All right, molecular defenses. Interferons are released by virus-infected cells, and they help prevent neighboring cells from getting infected by viruses. The complement system is a whole huge cascade of more than 20 proteins. It's a general system, and we'll talk about it here in a minute. And it's these proteins are made in the liver. And then there are antibodies. Now, there are many different classes of antibodies, but there are non-specific antibodies. And these are antibodies which are made against pathogen-associated molecular patterns, PAMPs, right? We talked about DAMPs already, but PAMPs are patterns of molecules which are found only in pathogens. And your body has learned to make antibodies to these and help neutralize potential pathogens in a generalized way. So interferons, we're not going to talk about a ton, but they can be used as an antiviral therapy and in other diseases, sometimes in cancers. You can use gamma interferon and things like that. There are several different types of interferons, but These interferons, again, are warning phone calls to certain cells in the body that protect them or activate certain pathways. Right? So for example, a virus can infect a cell. This cell recognizes that it's infected by a virus, and before the virus can finish its replication cycle, it will secrete interferon. And this cell will secrete interferon, which will then bind to receptors on neighboring cells, which will prepare those neighboring cells for an incoming viral threat, right? And this cell can then be better prepared to defend off the virus, right? This doesn't always work, and sometimes the virus overwhelms this, which in that case you would get sick. Now the complement system is a huge set of proteins here, and they can act by themselves. So these proteins can act by themselves, or they can actually be activated by specific immune responses. Okay? So the complement proteins can, when acting by themselves, can target phagocytosis by acting as something as what we call an opsonin. So opsonization is what your body does to a molecular or to a pathogenic invader. To kind of what it kind of does is paints molecular bullseyes on this pathogen and says, this thing is bad, eat it, right? So some of these complement proteins can opsonize pathogens when they bind, right? Antibodies can also do this. Right? Some of these complement proteins, when they're activated, can increase the inflammatory response and activate certain immune responses. And some proteins of the complement cascade can destroy invaders directly. There are several pathways of complement activation, and we're not going to get into the major details here, but there's a classic pathway and an alternate pathway. But whichever way complement is activated, right, either classical or alternatively, it leads to the same thing. Activation of the complement proteins C3 and C5, which then go on to hopefully help kill these particular invaders. Right? The MAC attack, which is the membrane attack complex, is a huge complex of complement proteins which actually can get stuck in the membranes of foreign invaders, right, and cause all these little holes. Right? So this is a bacterial cell that has the MAC attack stuck in their membrane here, and that causes the cell to leak, which then causes the cell to die. So when the MAC gets you, you die. Don't worry about that. So this is an overview of the non-specific components of the immune system, first line and second line defense mechanisms. So again, this is a lot of information, and I really just want to give you guys, or I want you guys to take away from this, a general overview of how your non-specific immune system helps keep you safe, right? And how these different things work together within your body. This is a very elegant system that works together at the molecular and the genetic level to help keep you safe. And this type of immune system is found in all across the animal kingdom, right? So it's not specific to us. Remember, we're not the only animals that get sick and die and have bacteria constantly invading us. Pretty much all multicellular life, and even bacteria, have a very remedial immune system. Right? So as always, if you have any questions, let me know. I'd be happy to answer them. All right? Have a good day.